Good morning, my dear student. I am Dr. Kiran Ramrao Borkar. Now today I am going to discuss about the urea cycle. Now what is urea cycle? The urea cycle is the first metabolic pathway to be elucidated. The cycle is known as a Krebs-Hensley urea cycle. Why it is called as a Krebs-Hensley urea cycle? Because the cycle was described by Krebs-Hensley. Ornithine is the first member of the reactions. It is also called as a ornithine cycle. Why? Because ornithine is the first member of this reactions. Urea is synthesized in liver. What is the site of urea cycle? That is the liver, and transported kidney for the excretion in urine. The two nitrogen atom of urea are derived from two different sources, one from ammonia and other directly from the alpha amino group of aspartic acid. Carbon atom is supplied by CO2. Urea is the end product of protein metabolism. What is the end product of protein metabolism? That one is the urea. Urea synthesis is a five-step cyclic process with five distinct enzymes. The first two enzymes are present in mitochondria while the rest are localized in cytosol. Two enzymes present in mitochondria and while rest in cytosol. Now we see here the steps of urea cycle. Look at here. This urea cycle occur partially in mitochondria and partly in cytosol. Now we see here the first step of urea cycle, and that first step is that the formation of carbomoyl phosphate. What happens here? Condensation of ammonium ions with bicarbonate ion resulting in the formations of carbomoyl phosphate. By help of the enzyme carbomoyl phosphate synthetase 1 present in liver mitochondria. Liver mitochondria, it requires Mg2 plus ions and dicarboxylic acid, and that dicarboxylic acid is that the N-acyl glutamate. This step requires 2 ATP. This step requires 2 ATP. Now we see the second step of urea cycle is that the synthesis of citrulline. Carbomoyl phosphate form in the first step combines with ornithine resulting in the biosynthesis of citrulline added by the enzyme citrulline synthase or ornithine transcarbamylase. Citrulline is easily permeable to the mitochondrial membrane and hence it diffuses into the cytosol. Now we see here the third step, that is synthesis of argininosuccinate. In the cytosol, citrulline combines with amino acid aspartate forming argininosuccinate. Catalyzed by enzyme argininosuccinate synthetase. It requires 2 ATP. It requires ATP which is hydrolyzed to AMP, resulting in utilization of two high energy bond. Mg2 plus act as a cofactor. Now we see the fourth step of urea cycle is that the cleavage of argininosuccinate. Cleavage of argininosuccinate. The enzyme Argininosuccinase act reversibly to cleave argininosuccinate into the arginine and fumarate. This fumarate enters the TCA cycle between the what is TCA cycle? That is the linkage between the TCA and urea cycle is known as a Krebs bicycle. Now we see the fifth step of urea cycle is that the cleavage of arginine. Cleavage of arginine. Arginine is a lyse into ornithine and urea under the influence of enzyme arginase. Hence, arginine is also known as a semi-essential amino acid that is through it is synthesized in the body is not available for protein synthesis. Ornithine is regenerated in this step as the urea cycle completed by the formation of urea. Ornithine and lysine are potent inhibitor of arginine. Enzyme enzyme arginase. This arginase is also present in testes, renal tubules, mammary gland, and screen in minute quantities. The intermediate, the intermediate amino acid form in the urea cycle that is ornithine, citrulline, argininosuccinate are known as a non-protein amino acid. Which one? Argininosuccinate, citrulline and ornithine are known as a non-protein amino acids. This is all about the urea cycle. Thank you so much.